Hi there. My name is Dr. Jocelyn Smith, and I'm an associate professor at the University of Guelph, Ridgetown campus in field crop entomology. And today I'm going to be talking about European corn borer, an old pest that's been around for a long time, but may become a new one again, or is having a bit of a resurgence because of some Bt resistance that we've discovered in Canada. It was introduced to North America in the early 1900s from Europe into the New England states and even into southwestern Ontario. We think it arrived on broom corn that was brought to this part of the world at that time. And it's pretty much expanded to all of the regions in North America where we grow most of our corn. You can see on the map on the top left corner of your screen the distribution of European corn borer currently in North America. And it's interesting to note that depending on the latitude of your location, we have multiple generations of corn borer. So in southern Canada, we have 1 to 2 generations per year, but as you go further south into the US, you could have 3 to 4 generations. And the graphic on the right kind of shows what a 2-generation population or a bivoltine population of European corn borer would look like. So the overwintering stage of the corn borer is a 5th instar larva, which is the largest larval stage, and they overwinter in the corn stubble within the corn stalks. In the spring, they pupate in the stalks, and then the moths leave those plants, fly around to find places where they can meet each other to mate, which is usually not inside a cornfield. It's usually grassy areas around cornfields. And then they look to find cornfields where they can lay their egg masses. Once the eggs hatch, these little caterpillars will get into the corn plants as quickly as possible. And so they'll do a little bit of leaf feeding. They might feed through the whorl, and we'll see some of the shot hole injury that can cause later on in some slides that I have. But as the name implies, they're going to spend most of their time inside the stalk, boring up and down, making tunnels. And yeah, it can go through multiple generations per year, depending on where you're located. So the issues with European corn borer, of course, as it's boring in the corn stalk, is that we get yield loss, right? The translocation, water uptake, etc., in the plant is disrupted by these tunnels that the corn borers make. The stalks become weak at different points, and then they can break. You'll get lodging of plants. You might get ear drop as the larvae might bore into the shank of the ear. And because the corn borers go in and out of these plants, or they do have entry holes on the outside of the plants, this allows entry points for more pathogens, which can lead to stalk rots, ear rots, and mycotoxins in grain. So those are all the issues that come along with corn borer. The yield loss quantity is around 2 to 6% per plant with 1 larvae per plant from some old research that was done in the Corn Belt. It's interesting, as I mentioned, this corn pest has been around for a long time. We have a lot of old extension documents about corn borer from the past. We've even had legislation in some parts of the Corn Belt where growers were required to destroy the overwintering habitat of European corn borer, which, as I mentioned earlier, is the corn stubble. And so that back in the day was the best way to control European corn borer. But over time, we've had some significant advances in terms of corn borer management, that being transgenic or Bt corn. So 1996 was when Bt corn was first commercialized, and the target pest was European corn borer back then. Cry1AB was the first Bt protein commercialized by Northrop King Monsanto at that time. And over time, we've had additional Bt proteins developed for corn borer, such as Cry1F from Dow AgroSciences that came out around 2002. And in 2008, another couple proteins, Cry1A.105 and Cry2AB2, which have been pyramided together by Monsanto, are the traits in VT double pro hybrids. So nowadays, the majority of the Bt hybrids that we plant in Canada and the US contain a pyramid. So at least two of these Bt proteins targeting European corn borer. The other above ground Bt protein that's not listed here is VIP3A, and that one actually does not have any control of European corn borer. So it's always paired with some of these others that do. Since that technology was commercialized back in 1996, it's been widely adopted in North America. And both in Canada and the US, about 85% of our corn nowadays that we plant contains Bt proteins that target corn borer. Before Bt corn was available to growers, European corn borer management cost about $1 billion a year for growers in North America based on yield losses and the cost to control them. But we think that Bt corn has been pretty much one of the best success stories in pest management in North America. And that's partly because we've had these high dose proteins, those 4 Bt proteins that I mentioned, that we call them high dose because they are basically killing more than 99% of the European corn borers that are exposed to them. The corn borers are very susceptible to these proteins. We've also had success in using our insect resistance management plans that included refuge over time. Initially, when we were planting single toxin Bt hybrids that only produced one of those proteins at a time, you were required to plant a 20% refuge. But as we've been able to pyramid those Bt proteins, we've been able to reduce the refuge down to 5% because we were having such good control of European corn borer populations. And we also moved to refuge in a bag to make it easier for growers to implement. So all of this was working really well. And as resistance monitoring had been conducted since the beginning of Bt corn deployment with European corn borer, there were no cases of Bt resistance developed in the field being detected before 2018. And so this is why we're talking about this again, because we've had an issue develop in Eastern Canada with European resistant corn. And this happened in 2018 near Truro, Nova Scotia. So we got some calls that year that we should go out and take a look at corn in Nova Scotia, that there was corn borer injury. We went to a number of fields that were heavily infested with corn borer. They were anywhere from 30 to 70% of the plants had symptoms of corn borer injury. And these were all Bt plants that produced the Cry1F protein. And so here on the map, I hope you can see this is the map of Canada on the left. There's a blue square down there at the bottom, which shows the majority of our corn production in Canada, which is in Southern Ontario, the province that I'm located in. However, where we found this problem with corn borer resistance was out in the Atlantic provinces, particularly Nova Scotia. And so I've zoomed in, if you follow the red arrow up to the map on the right corner, to the province of Nova Scotia and where the situation was initially found. So we collected corn borers from those fields, from 4 different Cry1F fields in that area in 2018. And we also collected a population to the west out in the Annapolis Valley that we thought would be upwind. So if there was Bt resistance in the populations to the east, they probably wouldn't be over to the west if they were going to spread. We collected a population from a non-Bt field there. And we brought these back to our lab at the University of Guelph, Ridgetown campus, and we tested them using our diet overlay bioassay, where we look at different concentrations of the purified Bt Cry1F protein, and we look at how what percentage of the test subjects die within a 7-day period. So you put these newly hatched larvae on the diet that's got the protein on it, and you measure mortality after 7 days. And so you can see in that graph, we have 2 populations on the left, Winger and Delaware from Ontario, that were all dying with really low concentrations of Cry1F protein. But all 5 of those populations we collected from Nova Scotia were almost unaffected by the protein. And so we can estimate LC50 values, or the concentration of the protein that kills 50% of the population, using these data. And the susceptible population LC50 values were around 6.5. nanograms per centimeter squared of Cry1F protein, but all of these populations from Nova Scotia, we couldn't kill them with 200 nanograms of the protein. So we consider these to have a resistance ratio, meaning they're at least 30 times more resistant to the Cry1F protein than the susceptible strains. And in previous experience with BT toxins and other pests, we would say anything with a resistance ratio over 10 is highly resistant. So this was the first time that anyone discovered a field populations of European corn borer with BT resistance. What the response was to this discovery was that, well, first of all, we learned that there were still being a lot of single toxin BT hybrids being planted in that region, meaning Cry1F only in this case. So those hybrids were removed from the market and they were replaced with a pyramid of Cry1F and Cry1AB hybrids for the most part. We increased our resistance monitoring across Canada because of this outcome, and we're trying to figure out a lot more about the corn borer populations in the Atlantic provinces to determine what else is different about them and why did this happen. So since that time in 2018, when we found the first resistant populations, we found more of them, unfortunately. So anywhere on this map where you can see these orange points, the green ones are still susceptible, but the orange points here show resistant populations. So we found quite a few throughout Nova Scotia and some up into New Brunswick and yes, in the eastern Canada there. We found one in Montreal, Quebec, or near Montreal. And we also found one way out west in Winnipeg, near Winnipeg, Manitoba. And so we've continued to monitor populations in that region from where the original resistance was found. And we were able to find a population that has developed resistance to Cry1AB in addition to Cry1F from that original spot. So that's unfortunate. And I would say that in addition to that, we have detected a lot of the Cry1F resistant populations we're finding in Eastern Canada are also becoming resistant to Cry1A.105 protein, which was one of the newer ones I mentioned earlier. So that's the Canadian story. Things seem to be getting a little bit worse in Eastern Canada, more proteins being compromised. And in 2023, by using some sentinel plots, where we put out BT and non-BT hybrids to monitor for resistance, this is a project that was led initially by Galen Dively from the University of Maryland. You can see these spots on the map where these sentinel plots have been placed in Eastern Canada. Another collaborator in Hamden, Connecticut, also planted one of those plots in 2023. And so here's one of our worst site in 2023 was from New Brunswick in Canada. And here you can see these bars in the graph show the number of holes per plant by the European corn borer. And you can see that there's corn borer injury in all of the different trait combinations that were planted at that site. And a similar thing was observed in Connecticut at that site in 2023. The pressure wasn't as high, so the injury wasn't quite as bad, but that's basically the first time we've detected European corn borer injury to BT plants in the US. So we've got issues there with Cry1AB, Cry1A.105, and Cry2AB2. So just to summarize where the resistance monitoring is now, basically we have detected resistance to Cry1F in Nova Scotia from 2018 until 2024. We found it in Quebec in 2019 and in 2024. We found it in Manitoba in 2020 and in New Brunswick in 2024. And in some of our field sentinel plots, we've observed that unexpected injury, so actual field injury to Cry1F in Nova Scotia from 2018 to 2025, and in New Brunswick in 2024 and 2025. For Cry1AB, we have only detected resistance in one population we found in 2022 so far, but we have seen unexpected injury to Cry1AB plants in New Brunswick in 2023, Connecticut 2023, Nova Scotia and New Brunswick in 2025. And then the last pyramid of Cry 1A.105 and Cry 2AB2, we have detected resistance to Cry 1A.105 in Nova Scotia from 2022 to 2024, and in New Brunswick in 2023. And we saw injury to Cry 1A.105 plants and 2AB2 plants, because they always come together, in New Brunswick in 2023 and Connecticut in 2023. But I say we haven't confirmed resistance to Cry 2AB2 using the bioassay method yet in Canada. So this is, you can see that a lot of 3 out of 4 of these BT proteins have become compromised in Eastern Canada and somewhat in Connecticut as well. So what is the problem with this? Well, the potential impact of corn borer resistance to BT corn is that our corn borer populations in Canada and the US may make a bit of a comeback. We have really good documented studies to show that we have suppressed European corn borer populations across most of these corn grower regions in Canada and the US because of using BT corn. And that's been a huge benefit economically to corn producers, and not just people who use BT corn, but there's a halo effect of this, so that organic or sweet corn producers that aren't using BT corn are also seeing reduced corn borer populations and less economic impact of this pest. So if we lose BT corn, this puts a lot more of our agriculture in jeopardy. We may see more yield loss come back because of European corn borer, and it increases our risk of mycotoxin contamination in our grain, as I mentioned earlier. We might see increased insecticide use as kind of a last ditch attempt to knock down some of the corn borer populations. But because of the behavior of corn borer and that they are usually quickly protected inside the plant, insecticide timing is really difficult, and it's maybe not the most economic approach. That's why BT corn works so well. There are other host crops for European corn borer as well that could also see an increased risk. Corn borers actually will infest over 200 different plants as their hosts. So there are a lot of vegetables that could be susceptible, fruit and vegetables that could be susceptible to corn borer if the populations come back. And then this also could threaten the use of foliar BT products, because some of the foliar BT products contain the same Cry proteins that we have in those transgenic options. So what do we recommend growers do at this point in time? Well, we wanted to bring this topic to your attention because it's kind of an important one to keep a lookout for. We want you to start looking for European corn borer again, especially in single toxin BT hybrids. There shouldn't be very many of those out in the marketplace. Probably the majority of hybrids we're growing are pyramid hybrids, so that the risk of seeing any corn borer injury to those is a lot lower. But if there are pockets where these single toxin hybrids still are being grown, we definitely want to have a look for those. And we're also kind of re-educating growers about what corn borer injury looks like, because we've controlled it so well for almost 30 years that most people haven't even seen this insect before, maybe in their lifetime, or they haven't seen it in a long time of their farming career. So we're going to go over some of the symptoms of the pest injury in a second. And where we have these areas with resistant corn borer populations starting to show up, we're talking to those growers a lot about how to suppress, mitigate, or control these populations to slow the spread of BT resistance. And I'll get to that again in a second. But so just to quickly go over a few, things to look for with European corn borer. This is what the caterpillar looks like inside the corn plant. If you split a corn stalk open, this is the caterpillar that you might find. They're they have a cream colored body, a black head. They have these spots along their body. They really don't have any distinctive stripes, but they do have these little dots along their body that are black, that have a black ring around the outside and a pale center. So we say they kind of look like a halo. And a fully grown corn borer larva is about 1 inch long, or 2.5 centimeters. The moths are really hard to see. They're very small. You can see they're triangular in shape. They're cream colored. they are sometimes found in cornfields, but they're sometimes found around cornfields. So if we might look into pheromone trapping, that's what the moths look like there in those photos. The egg masses are also quite difficult to see. They're small, they're white, they look like little overlapping fish scales, and they're usually laid on the underside of the corn leaves. But as I said, they're quite small. So I say if you think looking for Western bean cutworm egg masses is hard, looking for European corn borer egg masses is even worse, I'm afraid. Early injury by some of the larvae could look like little holes that you might see through the tassel, through the anthers into the stalk. And one of the telltale signs is that you see this sawdust kind of on the outside of the stalk, and that's what that is the corn borer's frass. So they've been eating and they're excreting that, and they're pushing it out of their little tunnels to keep their tunnels clean inside the plant. That is a really good sign of corn borer injury, and it could be anywhere on the plant, top to bottom. Later in the season, as the corn borers tend to go lower on the plant, again, you'll see those stalk or holes on the outside of the stalks and the frass that's being pushed out of those holes. That looks like sawdust. You might see broken tassels, broken stalks above the ear or even below the ear later in the season. And often the broken part of the plant might turn kind of red or purple discoloration from the corn borer injury. And lastly, there's a picture of one that's even bored into the shank of the ear, and you can see that the ear can drop because of that. So as I mentioned, an important part of managing corn borers is to understand that later in the season, as we're getting closer to harvest and into the winter, they go to the bottom of the corn stalks to overwinter. Some research that was done a long time ago in Canada showed that the majority of the larvae were within the bottom 30 centimeters of the stalk. And we've even seen in recent years in some of these areas where we're finding resistant corn borers that they are really down low in the plant, almost at ground level or below ground level in the base of the stalk. So one thing that we're encouraging growers to do in areas where we have resistance is to do some stalk chopping again, which not everyone has this equipment available to them, but it is probably going to help in controlling corn borer populations if you can do some mowing of corn stalks. But we're looking to do some more experiments and look at different tillage implements nowadays and what kind of mortality we can get of corn borer populations. So lastly, this is just to kind of recap. We've started to see resistance to European corn borer to BT corn in Eastern Canada and in the Eastern US now. And the corn borer BT corn story has been super successful for nearly 30 years, but as is usually inevitable with our insect pests, they eventually find a way around it. And so it's starting to possibly break down now. So we want to keep our eyes out for this problem. We want to determine the scope of the problem, where it's actually happening, and maybe mitigating the populations by controlling them somehow to slow it down, spread of resistance. We have a lot of research going on to characterize these BT resistant corn borer populations based on their genetics, learning more about the biology of the pests in these certain areas. There's a lot of questions around that that may help us manage them better. And frankly, we've kind of lost track of corn borer research since, as I said, we've controlled it for so long. There's a lot of things that we assumed about corn borer from 30 to 50 years ago that could be different, that we need to study now. And then we're looking at how we can manage them by monitoring for it, maybe using stalk chopping and things like that as cultural practices to slow it down. And potentially there will be some new BT proteins, or not just BT, other proteins coming in the future that we can use for corn borer. But again, we need to know more about the populations nowadays that can help us preserve any new tools that come along for a while as well. So lastly, I'd just like to thank all the people that have helped with the project. It's been a really big effort, and we've got a lot of really great new collaborations with people in Eastern Canada and in the Eastern US that have helped us with this.